Thank you for attending and welcome to the 2024 Postgrad Seminar. Before we begin, I'd like to make an acknowledgement of the country. So I acknowledge the traditional owners and the custodians of the land on which we are meeting here today, the Wurundjeri people of the Kulin Nation. I pay my respects to the elders past and present and emerging, and I embrace their continued connection to this place. So today, it's very exciting to introduce you Dr. Jovana Maksimovic, who is a senior researcher in bioinformatics and computational biology in the Oshlak lab at Peter MacCallum Cancer Centre. Her work has primarily focused in development in epigenetics and transcriptomics, data analysis and methods development projects, with a particular influence in the field of methylation array analysis. Jovana currently holds a NHMRC EL2 Investigator Fellowship and is leading the computational analysis for the development of a globally accessible cell atlas of healthy pediatric airway, which is funded by the Chan Zuckerberg Initiative Pediatric Center Atlas Network grant. Before I hand the mic over to Jovana, I'd like to just remind that attendees are also invited to join the speaker after this session for light lunch at the Tapestry Lounge. All right, I'll hand you over to Jovana.
Hi, thanks for the introduction and thanks everyone for coming along today. So today I'm just going to be giving like a really big, broad umbrella overview of methylation and how we measure it and then how we analyse that data. So briefly, I'm just going to zoom all the way out and talk a little bit about epigenetics and where DNA methylation fits in there. And then sort of zoom in on what it is and then how we measure it with sort of two sections, one focused on arrays and one on methylation sequencing. So what is epigenetics? Well, so one of the definitions is that epigenetics refers to stable heritable traits not explained by changes in DNA sequence. And the Greek prefix epi literally means on top of genetics. So we're talking about all the layers of things on top of the DNA sequence that help regulate gene expression. So these are all the different chromosome modifications you may have heard of, like histones, and of course DNA methylation. Now all of these elements are really essential for normal development, but they can also be modified by the environment, so they're not as sort of set in stone as the DNA sequence, and they can be disrupted in processes like disease. So because of this role in development and differentiation and basically making cells what they are, epigenetics is really, really complicated. So there are all these different aspects. And so many different types of sequencing and array methods have been developed, particularly over the last 10 to 20 years, to help us measure all of these different factors. But I'm not going to talk about all of this today. I'm just going to focus on DNA methylation, which is just one layer of all of these sort of epigenetic marks. So what is DNA methylation? Well, if we zoom right into the DNA, when we talk about DNA methylation, what we're referring to is the addition of a methyl group to the cytosine of the DNA sequence. And this typically occurs in this CG dinucleotide context, and that is the most common context that we see methylation in. It does occur in other contexts, but I'm not really going to be touching on those today. So I'll primarily be referring to CGs, and we tend to call them CPGs. So the human genome contains about 30 million CPGs, roughly, but that's actually less than what we'd expect. It's actually only about 1% of the genome. And this will vary between different species. So different species will have different numbers of CPGs. And in addition to sort of being depleted to what we'd expect, CPGs are not evenly spaced across the genome. So they tend to occur in these clusters that we call CPG islands. And so if you look at this diagram here on the top, these little lollipops here representing CPGs, they tend to be clustered together, and associated with regulatory regions, like in the promoters just upstream of transcription start sites. The other thing we know about CpG methylation is that it tends to be spatially correlated. So two CpGs that are very close together in space will tend to have very similar or the same methylation status. And that's illustrated by this plot down here, where we've got the distance in base pairs on the x-axis and then the percentage of identical methylation on the y. And so you can see that relationship here, that CpGs that are very close together are very likely to be identically methylated. And as they move further away, that relationship breaks down and sort of is completely lost after about 500 base pairs. Now, the reason methylation is really interesting is that because one of its sort of main things that it does is it can regulate gene expression. And so if you look at this diagram on the left, it's representing a gene here in these green boxes, and the CpGs are represented by these lollipops again. And the white ones or the empty ones are unmethylated, and the black ones are fully methylated. And so what we tend to observe is that when CpG islands in the promoter are unmethylated, you tend to get increased gene expression. And when they're methylated, you tend to get decreased gene expression. And that's what this plot on the right is showing, where we have percent methylation on the x-axis and gene expression on the y. And this is just looking at one single CpG in the RAB25 gene. And each of these dots is a single sample. And so you can see this relationship quite nicely here, where for very low levels of methylation, we get very high gene expression and vice versa. Okay, so because methylation is so important in regulating gene expression, it's really key in the process of development. So, in going from gametes to zygotes to blastocysts, there are huge methylation changes that occur to help with development and then differentiation into different cell types. And it's involved in processes such as exon activation and genomic imprinting. So methylation, very, very important for all of these processes. And based on that, it's very much critical in determining cell types. So, we know all cells have identical DNA, but different cell types look and behave very differently, and methylation is one of the reasons why. So there's a paper published in 2023 that sort of built this huge atlas of DNA methylation across different cell types. And you can see the differences in methylation patterns in regulatory regions in this plot on the left. So quite different for different cell types. And you can see this sort of nice relationship inferred from it here, and this sort of cell type tree where you've got, cells that are more similar in their function and more similar in their methylation, and, cells that are quite different in their function have quite different methylation patterns because it's trying to switch on very different genes on and off depending on what the cells need to do. But as I mentioned at the start, it can also be disrupted in disease. And so methylation can be significantly altered in diseases like cancer, for example, where, for example, here, if we have hypermethylation, so more methylation in the CpG island at the promoter of tumour suppressor genes, these can get switched off. And conversely, what can also happen is that CpGs in the promoter of oncogenes can be hypomethylated, so less methylated, and then these can be switched on, and you can imagine the sort of downstream consequences of cancer development from that. But because of this, cancer can also be a useful biomarker. Methylation can also be a useful biomarker of cancer, and it's of particular interest in contexts such as liquid biopsies, where we all have sort of DNA from different cells that have died and sort of shedding their DNA floating around in our bloodstream. And that DNA also contains the methylation pattern from its cell of origin on it. And this is also true of DNA that's shed from tumours. And so people have found that it can be quite a powerful marker of the cell of origin of a tumour, and it can also help us detect tumour DNA in this context. And so lots of people are working on developing this as a biomarker for cancer screening or prognosis or even detection of minimal residual disease, so really, really low amounts of tumour cells floating around. I really love this example of how differences in methylation can completely change a whole organism phenotype, even when we have identical DNA. So these mice here are agouti mice, and their coat colour is controlled by this gene PS1A. And so the yellow mice here on the left, they have less methylation at this promoter, and so their coat colour is more yellow because of the change in gene expression, whereas these mice here have more methylation, the promoter of these genes, and so they're a darker colour. The really interesting thing about this is that this change is completely induced by the diet of the mother. So depending on what the mum was eating, this changed the methylation pattern, the promoter of this gene, and then you get different coloured offspring, even though they have completely identical DNA. Another real sort of neat example of this is in bees. So bee larvae have identical DNA, but depending on their diet and what happens to methylation as a consequence, we can either get queen bees or worker bees. So worker bees are fed on a worker jelly diet and tend to be more methylated and thus become worker bees, whereas queens are fed on special royal jelly and are overall less methylated, and that's how you get queen bees. So completely different phenotype, identical DNA, affected by basically the environment, so the diet. Okay, so methylation, very interesting and obviously really, really important, but how do we go about measuring it in the lab, and then how do we analyse that data? So there are lots of different methods that have been developed over the years to measure methylation. A lot of them are actually are different types of sequencing approaches, and they've been coloured here on this timeline by sort of the broad category. So these sort of bluey-green ones here are kind of affinity enrichment type protocols where there is some protein that tends to bind to more methylated regions, and then you sort of enrich for those and pull them down and sequence them. And then you kind of get this regional view of methylation. These green ones are endonuclease digestion methods, and they tend to be quite targeted at very specific loci, so not very genome-wide at all. And these yellow methods are the bisulfite-dependent methods. So these sequencing approaches are based on bisulfite conversion of DNA to detect methylation, and they were some of the earliest methods developed, and they're one of the most popular. But recently we have another type of sequencing that's very, very similar in how it works, but instead of using this bisulfite treatment, they use enzymes to convert the DNA. So lots of different options. And then there are some newer methods that have been, this method called 5-letter sequencing for short was published just last year. So it's using short-read sequencing to try and detect methylation in addition to genetic differences. And then, of course, we've got the long-read platforms such as PacBio and Nanopore that can also read methylation as they read the DNA sequence. But I'm actually going to start talking about arrays because that's what I've spent a long time working on. But oddly enough, in the methylation space, it's actually one of the most popular ways still to measure methylation, and it very much depends on your application, what you end up choosing. So what are methylation arrays? Well, they're these little glass slides that have probes spotted onto them, like, little DNA probes, and the probes target particular CPGs across the genome so that we can measure their methylation using fluorescence. And they were originally released by Illumina back in 2009, so 15 years ago. And the original arrays were only able to detect 27,000 loci across the genome, so compared to 30 million, not very many. But they've been scaled up over the years, and the most recent version, which was released last year, can now detect nearly 1 million base pairs across the genome. And they're mostly focused on humans, but there was a mouse array released in 2020. So the advantages of arrays are that they give you single base pair resolution, so you can detect methylation at every CpG dinucleotide that's probed, and it uses this bisulfite conversion process that is, quite familiar to a lot of people. But one of the main reasons that they're actually so popular is because they're relatively cheap. So compared to doing sequencing, you can do lots of samples using these arrays for relatively low cost. So they're really popular if you're doing large studies with many samples. You're not going to be doing whole genome bisulfite sequencing because the cost of that would be astronomical. Now, the disadvantage is that really you only get what you get. So the probes that are on the array, 1 million maximum, is still a relatively small subset of CpGs across the genome, and they tend to be focused in and around genes, which for some applications is really good, because if you want to stick to regions that we know how to interpret quite easily, then things in and around genes and regulatory regions are going to be the easiest to interpret anyway. So in some ways, that's kind of an advantage of the design, is that we can look at areas that we already know a lot about. So I've mentioned that arrays use bisulfite conversion, as do a lot of other sequencing approaches. So what is it? It's a chemical process that was developed and published by Sue Clark at the Garvan Institute in Sydney, nearly 30 years ago now. And it's essentially a process that sort of creates kind of SNPs in the DNA that we can then detect approaches similar to SNP finding. And so what happens is that methylated cytosines, so methylated Cs, are protected from bisulfite conversion. So when you apply sodium bisulfite to DNA, what happens is that Cs are converted to Us, so uracils, but if they're methylated, they're not converted. And then after PCR, basically all the Us are becoming Ts, and the methylated Cs are the only Cs that remain. And so this is how you can detect whether something was methylated or not, through this sort of conversion process. So how does this work in the context of arrays? Well, as with any experiment, you're going to start off with collecting some samples. Maybe you'll collect 2 groups that you want to compare in terms of their methylation. And then you extract the DNA. And then you apply sodium bisulfite treatment. So you convert the DNA before it goes on the arrays. So then your bisulfite converted DNA is what's applied to the arrays, and it's the bisulfite treated fragments that are going to bind to the probes. And so methylation arrays have these little DNA probes attached to these beads, which are detecting methylation at these, say, 1 million CpG sites across the genome. And you're looking for a match between the sequence, your bisulfite converted sequence, and one of these probes to detect methylation. And so when a probe binds, a fluorescently labelled nucleotide is incorporated, and you detect this fluorescence. And depending on which base is sort of added, that signal either corresponds to that base being, that CpG being unmethylated or methylated. And so you detect both signals in the fluorescence. And that's what our data is. It's this fluorescent signal. And that's what we then go on to analyse. So what methylation values can we actually get when we're looking at this kind of data? Well, if we think about an individual cell and we only focus on one single CpG, and we all have 2 chromosomes in our cells for this particular, corresponding to this particular locus, what the 3 possible scenarios are that this CpG won't be methylated on either chromosome. It could be methylated on only one chromosome, or it could be methylated on both. And so the values that we would expect are then 0, 0.5, or 1. But when we do this kind of experiment with arrays, we're not actually measuring individual cells. we're actually measuring a population of cells. So that whole, cell smoothie concept. And the value that we actually get is a continuous measurement somewhere between 0 and 1. So we don't see this 0, a half, 1. We get some continuous measure somewhere in there, depending on what's going on across all these cells. And then what we convert our fluorescence data to are a few values that we typically use for analysis or visualization. So the most commonly used and the most easily understood one is the beta value, which is your methylated fluorescent signal divided by the total signal, which is your methylated plus your unmethylated. So it's a proportion or a percentage of methylation, which is really sort of intuitive to understand when you're talking about a single position. You say, I've got 0.2 beta value or 20%, that's really easy to visualize what that means. And a beta value distribution typically looks like this. So you can see that most CPGs tend to be either fully unmethylated or fully methylated, and then we've got this sort of gradient in between here. But beta values aren't great for statistical analysis because they're constrained between 0 and 1. And so what we tend to do all our analysis on are M values, which is the log ratio of the methylated to the unmethylated signal. And so you can see the similar distribution there, but the tails go out to minus infinity and plus infinity. So much better statistical properties for doing analysis. And the two values are related to each other via a logit transformation, so you can quite easily, with a function in R, convert between the two of them, depending on what you want to be doing. And the data looks a bit like this. So if you've ever dealt with gene expression data or any other data, it's very similar. So you've got your samples in the columns, and then your CPGs are your rows. And so you can imagine that for, say, the newest methylation array, you'll have 1 million rows, nearly one for each CPG, and then you'll have as many columns as you have samples, and then this is what we work with in the analysis. So how do you go about then analysing this data? Well, Bioconductor is the place that will pretty much have all of the useful packages that you'll find for methylation array analysis. If you go to the Bioconductor website, and you go to the BioC views or the BioC views page, and you type in methylation array, you'll find 45 packages related to methylation array analysis, and they'll all be focused on different aspects, and some of them will be doing very similar things, but it's kind of pretty much a one-stop shop for doing analysis. And Belinda and I, so Belinda Phipson is a lab head here at WeHi in Bioinformatics, so we worked together and developed our own package for methylation analysis called MissMethyl, and that's available there in Bioconductor, and it's got a few different functions for doing different aspects of methylation array analysis. Now, another advantage of using arrays, if they're appropriate for your application, is that their analysis is actually really mature. We pretty much know how to analyse them and how to analyse them well, and the signal we tend to get from them is really robust. So, if they cover what you want to be covering, then arrays are something that you should sort of seriously consider because there are lots of advantages to using them. And sort of the analysis workflow roughly looks a bit like this, where you load your data into R to pre-process it and then clean it up to remove bad samples or bad probes. So you do some pre-filtering, and there are various packages that will do this. And then you can move on to your sort of normalization and statistical analysis, where you're trying to find your answer to the biological question that you're interested in. And there are different methods available for doing this, and again, it will be guided by your application. And in the end, a very useful thing to do with methylation data is to try and look at it in its genomic context. So what's going on? Is it near a gene? Is it near some known regulatory regions? What's going on? And there are lots of useful packages and functions for that. And a handy way to sort of get started with this, if you're brand new to it, is to check out a workflow paper that Belinda and I published quite a few years ago now. But it's up on F1000 Research, and it basically guides you through the whole process, starting with your raw data and all the steps that you go through to sort of do an example analysis, and it includes code and sort of examples of what the output should look like and things like that. So it's actually a really popular resource that people, go to get started and then work with their own data. So I'm just going to quickly take you through what one of these examples looks like. And it's an example a real-life example that we published with collaborators here at WeHi, Len, who I see in the audience here today. And it's looking at differences between different cell types. So we published this paper with Len and Yusha here at WeHi all the way back in 2013, so more than a decade ago now. And the study looked a little bit like this. So our collaborators took blood samples from 3 male individuals and then sorted out some T cells and activated some of them, and then extracted their DNA and ran it on methylation arrays. And they were interested in looking at methylation differences between these different groups of cell types. That was what their focus was. And so I got that data and did all the sort of data cleaning and QC. And at that point, a really handy thing to do, and if you've done gene expression analysis, you've probably done this as well, is to look at the data and try and figure out what the main sources of variation are. And so what I'm showing here are 2 MDS plots from this study looking at the 4 main sources of variation in this data, so dimension 1 and dimension 2, and dimension 3 and dimension 4, and each of the sort of points are our different samples. And so you can immediately see that the largest source of variation in this data set was individual. So so the biggest difference was between the different people that the samples came from. But then if we drill down further into the principal components, we can see that now things start grouping by the different cell types. And so this is the signal that we're after. But these plots are telling us pretty plainly that we need to take this into account to sort of be able to drill into this. And so what we did then is to fit some linear models using Limma, which was published by Gordon Smythe here at WeHi. And the sort of patterns we're trying to identify are this. So we're looking for differences in mean methylation at individual CPGs between our groups that we're interested in. So in this case, our different cell types. And so what we did was we fit these linear models, including a factor for individuals to sort of account for that, and then also for our different cell types. And then we fit one for each of our million CPGs. And because we're doing so many tests, we have to adjust for false discovery rate. And at the end of this whole process, we actually got, depending on which cell types we were comparing, hundreds or thousands of differentially methylated CPGs, which can be a bit hard to interpret. Like, what do you do with a list of 1,000 differentially methylated CPGs? What does that mean? What is that telling you about the biology that you're interested in? And so in the gene expression world, the thing people tend to do is do some pathway analysis or gene set testing and try and figure out, what things are going on functionally, what processes are these differentially methylated genes representing. But the problem we have here is that we're not actually measuring genes, we're measuring CPGs. And how you relate them to genes is not exactly straightforward. So you can see that there can be a one-to-one relationship between a CPG and a gene, but it's usually a one-to-many, and sometimes it's a one-to-none. So what do we do here? How do we do our gene set testing in this context? And people did try and do it. People 10 years ago tried to apply gene expression methods just straight up on methylation data, and things went very badly. So a whole paper was published on how biased the results were and how you shouldn't be doing this. And so what Belinda then did was look into these biases and try and characterise them and to develop a method that does this in an unbiased way to help you interpret some of these results. So she developed this method, GOMETH, and the 2 main biases she found were affecting this data was one, which was pretty obvious, and it's probe number bias. And so you can see in this diagram that different genes have different numbers of CPGs associated with them. And because of this, they'll also have different numbers of probes measuring them. And this introduces this bias where a gene that has more probes is more likely to be found differentially methylated than a gene that has only 1 or 2 probes. And so you're automatically biased to the genes with more probes. So that's one big source of bias. A second source of bias was this, what we called multi-gene bias, in that we found that about 10% of all the CPGs were actually annotated to more than one gene. And so sometimes you would get CPGs associated with entire gene families, and these gene families would tend to be, they're related to each other functionally, and so they end up in the same Go category, and so they'd really inflate some Go categories. And if you happen to have these CPGs in your differential methylation results, then some Go categories tend to stick out like a sore thumb, but they're actually not biologically interesting. And so GoMeth is a method that accounts for both of these biases in various ways. And I'm not actually going to go into the details of the statistics, but basically, try to weight the results in different ways so that we sort of take away some of these biases so that the results that you get out are actually biologically meaningful and not just full of bias. But what about differentially methylated regions? So if you remember all the way back to the start of the talk, I said CPGs are spatially correlated. So CPGs close together tend to share methylation status. And so one signal that people like to look for are what we call differentially methylated regions. So patterns that look like this when you're, say, comparing two samples, where there's lots of CPGs close together that are doing similar things, but very different in different sample groups. And it's thought that if these regions kind of coincide with regulatory regions, that these are actually really functionally and biologically interesting, much more so than just one lone CPG that you might identify by itself. And so there are lots of tools out there for finding these kinds of differentially methylated regions. One of them that I've used a few times is Demarcate, which was also developed locally in Australia, up in Sydney by Tim Peters. And once you sort of run one of these tools, what can also happen, depending on the biology you're looking at, is, again, you're faced with a big long list of differentially methylated regions and you have no idea what this means. And so, you might think, oh, gene set testing. Let's see if, any pathways are pinging or anything like that. But you get this problem of bias again, because you can imagine if you're looking for a differentially methylated region, you can't actually find it in the first place if you don't have lots of probes in that gene or in that region to actually detect it. And so you're even more biased towards the genes and regions that have lots of probes. And so you have to take this into account if you're going to do gene set testing. And so we developed this kind of extension to GoMeth called GoRegion, where you can now do gene set testing in an unbiased way. And it sort of works a little bit like this, where you do your region finder, you identify some interesting regions, and the methods will spit out sort of a table of ranges where, you got a region here on chromosome 1 between here and here. And then you pass these into the tool. And what it then does is it basically scoops up all the CPGs annotated on the array in those regions and passes them to GoMeth for gene set testing. And it's actually really effective, even though the concept behind it is simple and really leverages what GoMeth already does. So what should you do? Well, what we found is that GoMeth is quite sensitive to the number of CPGs that you put into it. So if you've got lots and lots and lots of differences, these will map to so many genes that, you'll nearly cover all Go categories and then your results are useless. So we tend to sort of, rule of thumb, say, don't put in more than 5,000 if you're testing CPGs individually. But if your biology actually has so many differences, we actually recommend maybe doing region finding instead of testing individual CPGs, and then, applying something like Go region. Because sort of there's lots of evidence that you're going to be finding more biologically meaningful regions if you do it this way. And if you want to read more about any of this, more details of the statistics or benchmarking of these methods, you can have a look at our paper, which we published a couple of years ago in Genome Biology. Okay, so I've talked a lot about arrays, which are probably a little bit old school, even though they're very useful. So now I'm going to switch gears and touch on some sequencing methods, which basically you have to use for everything you can't do with these arrays. So here's this diagram that I flashed up before with the different sequencing methods that are available, and then some of their sort of pros and cons, advantages or disadvantages. Some of the methods, like the endonuclease digestion and the bisulfite-dependent bisulfite-free, give you single nucleotide resolution. So you can identify methylation at individual bases. But these endonuclease methods tend to be very site-specific, so you're not going to do that genome-wide. Whereas these methods, the bisulfite-dependent, bisulfite-free, you can use targeted panels, but you can also go genome-wide. And again, affinity enrichment methods, they're lower resolution, so we're only looking at methylation across regions, not individual CPGs. But for your application, that could be okay. And you can do these at a large scale with relatively low cost. So ultimately, how you choose a method will sort of depend very much on your biological question and your application. Your sample quality and quantity is going to play a part, because some of these methods you can't use with very low input DNA. You need a lot of DNA. So if you don't have a lot of DNA, some of them completely just fall off the list. And of course, budget. That's always a consideration. So if you don't have a lot of money to play with, then maybe whole genome bisulfite sequencing is not for you. And if you're interested in sort of knowing the more nitty-gritty about the performance of these various sequencing approaches, the EpiQC study published by Christopher Mason's lab in 2021 does a sort of head-to-head comparison of a bunch of these different sequencing types on the exact same samples. And so this is a really useful resource for sort of helping you decide what you might want to use in your own experiment. Okay, so just before I finish up, I'm going to focus a little bit on these conversion type methods, as I like to call them, the bisulfite dependent and the bisulfite free, because they are the most widely used, I think, of the sequencing approaches. And the basic workflow that these methods take is really similar, which is why I've lumped them into one category of base conversion, where you get your samples, you extract the DNA, and then you apply some sort of conversion, whether it's bisulfite treatment or an enzymatic conversion. The steps are kind of very similar. And at that point, you would construct your library and do your sequencing. And so, yes, regardless of which method you're doing, you're converting bases so that you can detect methylation. And just to remind you what that looks like, you start off with your, methylated CPGs here, and then you do your conversion. Doesn't matter which type it is, you'll end up with, unmethylated Cs converted to Us. And then you do a PCR amplification, and you get these sort of 4 strands out of your fragment. And depending on the kind of library that you generate at this point, you can actually sequence all 4 of these and then go on and do downstream analysis. But there's some important considerations with this, because you're reducing the complexity of your sequence by essentially converting one base to something else, and it makes it harder to map and a bit harder to analyse. So there are specialised methods to do this. You can't just use a regular aligner. But luckily for you guys, there's a thing now called Nextflow, where people have used this system to basically develop sort of open source pipelines and make them publicly available. And so When you get your data off the sequencer and you want to map your reads and call your methylation, you can jump on the NF-Core website and download this methylseq pipeline, which gives you two different aligner options for mapping and analysing this type of data. So Bismarck, which was one of the original mappers developed for mapping bisulfite converted data, or BWA meth with this other tool, methyldactyl, which is a different mapper, and then combined with this tool to actually call methylation at different sites. So Bismarck's been around for a really long time, but I think these days it's considered a bit slow and clunky, so I think most people these days tend to use the BWA methyldactyl option. And regardless of what type of conversion method you use, you can run these pipelines, so that's a really handy way to get started. And the basic pipeline sort of looks like this, where you've first got to create your genome reference, which for converted genomes particularly, well, it's important for all genomes, but you've got to do it in a special way for converted genomes. Then you do some, QC that's built into the pipelines, and then you've got to trim off all your sequencing adapters and things, and then you get to the point of aligning your reads. And once they're all aligned, that's when you can actually try and pull the data out. So you can actually try and call methylation at various positions across the genome. So what does that look like? Well, if you can imagine, you've got all your reads aligned to the genome, and you've got all these CG denucleotides everywhere. You would then focus in on one, and then you literally count up how many Cs do I have at this position, because they represent CPGs that were methylated, whereas any that are Ts at this point were unmethylated to begin with, and so were converted. And so you count them up and divide by the total coverage, and you get your percentage methylation at this base. Whereas if we look at this other base, you can see that there are fewer Cs, so at that base we've got 20% methylation. And you can imagine here that obviously coverage is really important, because you can't be really confident about how much methylation you call at a particular position unless you've sort of got sufficient coverage for it to be representative of your sample. And this is what that looks like in some real data. So this is data from the Broad on some tumour and normal samples, looking at a CPG island upstream of this STK33 gene. And so you can see that the blue here is representing unmethylated positions and the red methylated. And so you can see this massive difference here between the tumour and normal at this position. And this will be the sort of signal that you might want to detect in your upstream analysis of this data. You're not going to scroll through all these screenshots trying to find it. You're going to try and do some analysis to try and pick out all of these interesting regions. So the data that you work with looks a bit different to the array data. The data that comes out of the alignment and methylation calling looks like this. So you've got your position of the C in the genome. There will usually be the percentage methylation at that location. And then you will also have the number of methylated reads that have been called and the number of unmethylated reads. And by summing these two columns, you can get the total coverage at that position. So this is all the information that you need to go on and do your downstream analysis to try and identify interesting methylation patterns in your data. Okay, so what do you do at this point? Well, again, it depends. It always depends on your biological question. But just like with the example I showed with arrays, looking for differences is something that people quite commonly do. So doing a differential methylation analysis. And if we jump on Bioconductor, look for methylation sequencing tools, you'll find these 11, and these top 6 here have all refer to, containing functions and algorithms for doing differential methylation analysis using sequencing data as input. Another handy method that you could have a look at is based on EdgeR, again developed by Gordon here at WeHi. And there's a whole workflow for how to do that available at F1000 Research. So again, lots of valuable resources out there. But what about the fancy new technologies? A lot of the stuff that I've talked about, has been around for a while, but, everybody wants to do long reads now and other cool stuff. And so the good thing about that is that these newer platforms do natively detect methylation when you get, when they sequence your DNA. So this example here of nanopore sequencing. The way it works is your DNA goes through all of these pores, and as it passes through, it creates changes in the current that's going through here, and these little changes are then detected, generating what's called a squiggle plot. So it's this change in the electric current, and based on the shape of these, we can call what base is going through the pore at any time. Now, the neat thing about this is that methylated bases or modified bases change the shapes of these curves. And so you can basically, use software to detect when a modified base has gone through the pore compared to an unmodified base. And so the software to detect these differences in this signal is getting better and better. And so the calls of methylation in nanopore data are getting better. And so that is an option. If, long reads are something that it looks like you're going to need for your application because maybe, you're looking at repetitive regions or, you have some other requirements that just aren't great with to characterize with short read data. So that's always an option. There's also PacBio. PacBio sequencing can also natively detect modified bases, and so PacBio works a bit differently to nanopore in that you've got your DNA sample here, or your DNA strand, and then they attach these hairpins to it, which then go round and round and are sequenced by this sort of immobilized polymerase. And as each base is incorporated, a label base is incorporated by the polymerase, they detect the incorporation kinetics of the enzyme in real time. And again, they're looking at the shape or the distance between these curves to call the different bases in the DNA. And again, if you've got a modified base, this looks a bit different to the unmodified base, and so we can call this signal increasingly reliably. And one of the major advantages of, both of these platforms is you're not doing a lot of crazy prep to your samples, particularly in the case of nanopore. So this is just the final thing I'm going to touch on before I finish up, and it's this method that was published last year that uses short read sequencing to detect both methylation and SNPs in your data. So not a new sequencing method. It's just a really cool way of manipulating the DNA to try and capture more bases rather than just our 4 ACTG. And so if we go through this figure here that's from the original paper, we've got our DNA here that has got our methylated Cs up here. And if you just sequence it normally, you can only detect 4 bases. That's all you can get with short read sequencing. But we know that some bases are modified and we want to be able to pick that up. Now, what they came up with is this really neat idea of actually trying to use pairs of bases. So if you consider every possible pair of ACTG, you now have 16 combinations. And so the protocol tries to generate these pairs of bases and uses the conversion approach to convert unmethylated bases and then try and detect all 5 of them at the same time. So it's a bit crazy, but we'll get there. So we start off with the double-stranded DNA, which has got some methylated Cs here, which are coloured in green and yellow. And this is fragmented, and these hairpins are added to the ends. And then they're split. And both of these halves will go through this same process now. And so once these are split, one of your halves, the second strand will be synthesized, the complementary strand, and then some adapters get added on And then this conversion process will happen here. And some of these unmethylated Cs, again, will get converted to Us. And what you get up here is now a brand new fragment. So if you open it up with the hairpin in the middle, you've got sort of, the original half and then the sort of the synthesized half. And this is now the fragment that you end up sequencing from both sides using paired end sequencing. And so you're sequencing in from one side and sequencing in from the other side. And what you do at this point is you take those two reads, you don't align them to the genome, you align them to each other. And what that generates now are these pairs of bases. And there's this sort of table which kind of tells you what each pair actually means in the original sequence. And so if you see a CG pair, that means you had a methylated C to begin with. So it's a really, really clever way of trying to pick out methylation. It can also correct sequencing errors and obviously detect SNPs at the same time. So it's a very cool idea. And that sort of brings us to the end. So to sum up, methylation is pretty important and interesting, and we can measure it in lots of different ways. But the best technology for the job really depends on what you want to know, and you have to think about that quite deeply. What do you want to know, and then what the best technology is to measure it. Methylation arrays are still really relevant in this space, but they're only for human and mouse, although they're very cheap. But they cover limited CPGs across the genome. But the tech is robust and the analysis is very mature, and there's lots of different methods. So lots of advantages there if they fit what you want to be doing. Otherwise, there's lots of different types of sequencing, and it's pretty much your only option if you're looking at species other than human and mouse. And again, your choice is going to be heavily guided by your application, your sample type, and your budget. There are some existing pipelines to analyse the conversion type method data, and if you're using long read methods or like this 5-letter seq, there are sort of special pipelines that go along with them. But the downstream analysis in this space is still very much developing. And so, there are some methods on bioconductor that you can start with, but, it's still a little bit Wild West. So I'll finish up there. Thanks very much. Yeah, so the one space where people are really excited about it is in this liquid biopsy space, because obviously, tumours have a particular genetic signature and they have a particular methylation signature, and if you can detect both robustly at the same time, so at the level of quality that we're used to with short read, then that's pretty exciting. I think that's why people have gone on to develop this technique, because it's kind of ideal for these kinds of applications. So do you mean in terms of how you would measure it, or? Yeah, I mean, methylation can certainly change over time based on something that you're doing to your sample. But I guess all of these methods are a snapshot in time. So you would, with any of these methods, you would have to serially sample. So design your experiment such that you are, sampling the time course that you're interested in, and then you measure with whatever method at the time points that are relevant in your experiment. And then you can, compare them in various ways to sort of find differences between the time points using some of these statistical methods that I've described. Does that sort of make sense? Yep. I think methylation is actually one of the more reliable and stable signals in cancer. I mean, along with of the epigenetic signals. So along with sort of, DNA, which we're pretty confident in measuring the mutations in tumours in cancer samples, I think methylation is a pretty stable mark on the DNA. So it does contain that signature of the cell of origin. So I know our collaborators over at Peter Mac are actually really focusing on characterising methylation in some of their liquid biopsy samples, particularly for their cohort of samples with tumours with unknown origin, so where they can't classify it as a breast cancer or a kidney cancer or whatever, but they're now really looking into how to use the methylation to try and determine, where the cancer originated, because that signal is so tied to the cell type that it came from, and also the sort of the cancer that the way the cancer's sort of arisen. Yeah. Does that answer your question there? Yeah. I think that really will come down to sort of, throughput and cost ultimately. If these can get sort of low enough, I can see in the future that becoming an option for more applications. I don't know if it will ultimately, replace everything, and, that will be the thing that everybody does. But as you rightly point out, and I didn't really mention it on my talk, so this bisulfite treatment that I mentioned is really, really harsh on the DNA. And so and so it degrades the sample quite a lot. And so one of the reasons these enzyme conversion methods were developed are in that they're a much more sort of gentle process and don't damage the DNA as much. And so that is definitely a factor in there of why we're trying to move away from bisulfite conversion. But yeah, in terms of how much these long-read methods are going to take over, I'm not sure. They're going to have to get more accurate. So the accuracy is going to have to get on par with sort of what we expect for short-read and arrays. And, they're going to have to get cheaper to be able to apply them in sort of more cases rather than, I've got this like niche little thing that I want to try, which is what people are doing at the moment, to be doing these kind of larger studies. So, yeah, I don't know. I mean, Nadia's the nanopore expert, so...
So I think what's really nice about long reads is that you can phase the methylation, and there's lots of nice studies out there showing like silencing of whole, genes in large sections, which because you phase the separate genes. So that's one.
Yeah, so, but where is it, like, in terms of, like, cost? I don't generate any of this sort of data, so I don't know where it's at, but I know a lot of our collaborators are hesitant to go there because it's just too expensive at the moment.
Like, I mean, they're claiming it's as good and it's cheap, right? But companies want for they're not that high. But I don't know. I'm not an expert.
Yeah.
No, and I mean, the other.
Yeah. Yeah, it's a bit of this, like, you know, what can you do now, and then what will be done in the future? And I guess someone who wants an answer to their question now or within 5 years is still probably going to be using the current technologies for these reasons, that, you know, they're, you know, robust and we know how to analyze them and we can get the answers now. There are lots of these potential advantages with these newer technologies with long reads, but, you know, how long is it going to take for us to be able to analyse them really well and accurately? I don't know. That's a really open question. I mean, a lot's changed, say, in the last even couple of years with, you know, how accurate nanopore is. So, you know, if they keep building on things like that, maybe, but then that's only getting you to the point of calling the bases. What do you do downstream and how do you analyse this data downstream as well? None of that is really in place yet. People are just sort of doing ad hoc things based on their immediate application. So I think there's going to be a time lag. Might get there, but, you know, not next year. Yeah, no, that's a good question. That the spatial correlation I was referring to is specifically so in linear space. So the bases on the DNA, how close are they to each other in sort of that cis space? And so, yeah, the CPG island example where you've got lots of CPGs all really close together, they tend to all follow the same pattern. In terms of the looping of the chromatin and what happens then, I don't know. I don't think they're necessarily correlated, but I'm happy to be corrected if someone's actually looked at that sort of data and shown those kinds of relationships.